It's a pleasure to be here again with so many friends, and I have a difficult task today. First, I have to introduce the field which rapidly expanded in the past, I would say, 5 years into many different areas of neurodegeneration. And it's the field which, as a result, there is sometimes quite a confusion. So please be patient, and if you have questions, which I think inevitably follow, we will be happy to answer them during the roundtables and in the discussion during the coffee break and all the other functions which will continue here. What I will try to explain today, how those 4 pictures on the first slide are actually linked. The first one is the Kuru with a child with Kuru, which was the disease discovered by Guy Dussek in 1959 in Papua New Guinea, with pathology which really looked like a very old Alzheimer brain. So his first term for that finding was that it has to be something with very accelerated aging, despite the fact that this child was about 8 years old. So the later development through many work of many researchers show that it's actually a protein which is infectious and which is causing the disease and very rapidly progressive neurodegeneration. The other pictures I don't have to introduce: Michael J. Fox with rapidly progressive early onset Parkinsonism, Ronald Reagan with the Alzheimer disease, and Dave Derson, who was a very famous defense for Chicago Bears, who died in 2011 with chronic traumatic encephalopathy based on, again, rapid brain neurodegeneration. We have really epidemic of age-related neurodegenerative diseases. While the prion diseases we will hear today about represent a relatively small fraction, we see about 1 case per 10,000 deaths in US, with slightly higher numbers in age 65 to 75, 4 to 6 per million, and probably likely more carriers of the prions in UK because the exposure to the mad cow disease. There are many other diseases with very high frequency, like Alzheimer's disease, and we estimate that currently in the United States there is around 5 million cases of patients with Alzheimer's disease. And by age 85, we see incidence which is about 1 in 3. Then Parkinsonism, frontotemporal dementia, Huntington disease, chronic traumatic encephalopathy. So I think that the main goal of my presentation today is to raise the awareness of the fact that research in those diseases compared to the other diseases, like, for example, AIDS, 6 billion per year, is really underfunded. And this is summing all the research in all the Alzheimer's disease-related disorders. So we see about 5 to 6-fold difference between, for example, AIDS funding and Alzheimer's disease. So this, I think, is limiting the progress, which I believe could be much faster and much more efficient if there would be better support through National Institute of Health, and research would be funded more. Brain health, I think, is becoming a major issue which has a major impact on the society. And I think that it's becoming a major new frontier in the intersection of neuroscience, public health, and finally, social stability, because all the care of the patients is extremely demanding, not only on the families, but also on the infrastructure and public health funding. you will hear a lot of beautiful molecular biology today from best researchers in the prion field. Sometimes I think it's a good idea to repeat the context. So the context is human brain. The human brain contains about 86 billion neurons. Each of them has about 10,000 connections. So if you multiply it, the brain has about 100 trillion connections, which is about the number of stars in the Milky Way. You can imagine the complexity and the difficulties to study such a very complex and interacting system. We know that you need just a little bit of incorrectly shaped protein and that equilibrium, that fine-tuned system with those beautiful functions. And we have heard Valerie Sims playing beautifully, as well as we will see the researchers reporting the data that how that beautiful system can get destroyed very quickly. Now, you will hear about the prions. Prions effectively change shape from the normal, which is on the left, to the abnormal one, which is on the right. And this is our best representation of many data which Byron Coe put together in the most recent review on the subject. So there is a difference in shape. Now, how much you need of that badly shaped protein to destroy the human brain? You think about it as a large quantities, but it's actually only 7 to 15 milligrams of the prions to totally destroy human brain. Now, how much is 7 to 15 milligrams? I mean, it's about 1 grain of rice. 1 grain of rice can destroy human brain. So you see the difficulties we have. On the one hand, we have prions which are differently shaped normal protein, and on the other hand, we have these low levels which are extremely toxic and destroying the cells. Prions are forming those elongated amyloid-like structures, fibrils or rods. This is a structure which actually exists with many other proteins, including the Alzheimer amyloid. And it has very similar shape, and it looks very similar to the prions. So what we learned from prions? We know that prions and the prion protein, the pathogenic one, the toxic one, can form different shapes. And so basically not one shape is same as the other. And each of those shapes is related to the different toxicity, different replication rate, different ways how the disease develops. So all those different shapes, we call strains, are important to understanding of the disease. We know that they can even coexist, and then they select themselves into the most rapid and most toxic species. We know that they can evolve and exhibit the regulation of the substrate. So there is many cellular interaction which I think we will hear about. We know that the progression rate and phenotype of the prion diseases is the result of the shape, which is the molecular characteristic of the pathogenic prion protein, as well as the genetic background of the patient or the animal or suffering from the disease. So that interplay then dictates the specific features of the disease and specific characteristics. So the prion research was in forefront of asking all those important questions related to the genetics, what exactly happened in those interactions, how those different genetic polymorphisms which drive differences in the duration of the disease and some other aspects influence the characteristics, how the other mechanisms are important in the disease development if there is absolutely no nucleic acid which would explain those differences. So everything is related to the protein and shape of the protein. What is the origin and evolution of the prions? How it happened? I mean, how it happened in the sporadic cases in the absence of nucleic acid? What is the mechanism of the propagation and progression rate? What are the determinants of those variable progression rates? So we will hear from different investigators today in different aspects of those research goals we have. This is a artistic representation of what happened in Alzheimer brain. As well as in the prion diseases, in Alzheimer brain, you have accumulation of the abnormally folded protein, which accumulates into large deposits called plaques. Now, the cells around neuron cells suffer, and you see basically how they deteriorate due to the toxic effect of that incorrectly folded protein. In this case, it was amyloid discovered by Joe Glenner in 1984, who analyzed the content of those plaques. So that protein is formed through precursor, which is membrane-bound, called amyloid precursor protein, and by two proteases is clipped into the short fragment. That short fragment can misfold and form larger and larger aggregates, which then go to the big plaques, which are then seen, in the brain deposited, and it's a major marker of the disease. The progression starts usually in very small brain area, the hippocampus and surrounding areas. And from there, during the disease development, we see the spread of those plaques and the amyloid into the other brain areas until the brain is completely full with the deposits of the abnormal protein. Second aspect, which happened in the Alzheimer's disease, is effectively the railroad system transporting nutrients and the important molecules from the cell body to the other components gets destroyed. It's called microtubules, and it's serving as a railroad to transport many molecules. Now, one protein essentially for forming those railroads is tau. Tau protein seems to be essentially for assembling of those precise railroads transporting the nutrients and other molecules down into the important endings of the neurons, which are actually communicating with other neurons. So when it happens, you start to see that those filaments disintegrate, and that means that the cell completely stops functioning because the transporting the function is lost, and cells die through the process you see over here. We have found a very aggressive form of the Alzheimer's disease effectively by accident. We, as a Prion Center, we receive all the cases from all the United States with rapidly progressive neurodegeneration, because the assumption is that if it is rapidly progressive, it has to be prion disease. About 40% of those cases are actually Alzheimer's disease, rapidly progressive Alzheimer's disease. So when we compare those standard normal Alzheimer's disease cases, which have average survival time about 9 years, to our cases, we found out that they have survival only 7 to 9 months. So those are very malignant, aggressive cases of Alzheimer's disease, which are clinically very reminiscent, difficult to differentiate from prion diseases. So what is causing those rapidly progressive forms? We looked at the pathology, and we didn't see any difference. If you look at it, those are the deposits of the amyloid I told you about, which is the marker of the disease, and it was basically about the same level, the same distribution in all areas. So when we compare it, we didn't see any statistical difference between the cases with low or intermediate or high load of the amyloids. So in conclusion, I mean, in this part, we have, and not only us, but other Prion Center in Germany and in Japan already found that there is this very, very malignant form of the Alzheimer's disease, which features some deposits of the amyloid and tau proteins, which are indistinguishable at the standard pathology level from the other classical forms of the Alzheimer's disease. So the next question was, is it possibly related to the genetics? So the obvious high risk factors in the Alzheimer's disease is the high frequency of the apolipoprotein E, which is one of the cholesterol carriers with the E4 allele. Don't worry about the details, okay? But this is a marker of the high risk of Alzheimer's disease. And if you look at the frequency in our rpAD cases, it was about the same as in the normal population. So when in the Alzheimer disease, in the classical, you see very high frequency of this high-risk gene, you see very low frequency in those rapidly progressive cases. So we ask next question. Can we look at the plaques themselves and see what are the components? Maybe the plaques look different, the same, but they may be different. Composition may be different, may be composed of different proteins. So in collaboration with Tom Wisniewski at the NYU, we use the laser capture dissection. So you can, under the microscope, you can dissect the plaque, which is the accumulation of the amyloid, and then look at the composition by mass spectroscopy. We find out that the plaques in the rapidly progressive cases have totally different protein tracers than the slowly progressive cases. So effectively, they differ very significantly in many different tracer proteins. So that suggests that the plaques in the rapid cases is composed of proteins of different origin, mostly neurons. In the slow cases, it was mostly the glia. Now, this is, I think, the first important indication that the rapid Alzheimer disease may be actually a different subtype of the Alzheimer, and it may have different origin, different mechanism, and different pathogenesis, so effectively different disease. If you look at the other aspects, we also found out that the rapidly progressive cases have very high levels of what we call small oligomers. Those are not the amyloid plaques, but small aggregates of few molecules of the incorrectly shaped amyloid, which we think, and it's a consensus with the prion field and other aspects of other neurodegenerative, are probably most toxic because they are diffusible, they are mobile, they can move to the receptors and other molecules and trigger toxic responses. The other aspects which we found was that those molecules were differently shaped. Now, the way how we do it is, I will not go to the details, but basically each of those curves represent different shape of the pathogenic prion protein in sporadic CJD type MM2 or sporadic CJD MM1. And each of those curves is a marker of the conformation shape. And you see there is a spread values, and the red curves, which is the MM1 type, indicates differently shaped molecules, and we know that the MM1 is about 4 times faster disease than the MM2. So those differences in shape are related to the progression rate of the disease and the specific features of the disease. In the Alzheimer disease, we see in the amyloid very different spread and different shape of the curve. So in bottom line, the prion protein, prions are not amyloid. Amyloid beta has different conformation. And there is a big spread which we could relate again to the different types and subtypes of the disease with regard to progression rate. We were able to actually isolate some of those shapes and link them to the rapid progression, which statistically showed a very convincing correlation. So in this part of talk, I think the conclusion is that we have a disease, a subset of the Alzheimer disease, where the genetics is very important, but we have also different shapes of the amyloid beta, which is driving the progression rate and the disease characteristics. And different strains of amyloid beta may be as important as different strains of prions for the disease progression and disease characteristics. So in conclusion of this part, the broad spectrum of the distinct conformational structures of amyloid beta, but not the other form, argues for distinct prion-like strains. Correlation between those genetic findings and structural data, I think, are really important for understanding differences in the progression of the Alzheimer's disease and different phenotypes. I think that isolating those strains may help to develop better transgenic models, which would then be used for the development of therapeutics and new diagnostic protocols. We tested actually the rapid amyloid isolated from human brains and slow amyloid from the slow cases of the Alzheimer's disease in transgenics developed by David Westavey at the University of Alberta. So when we inoculated left hemisphere of the mice with the rapid extract from the rapid Alzheimer's disease brain, we saw the progression to the other side of the brain much faster than in the animals inoculated with the slow Alzheimer's disease extract. So in effect, we were able to show that the rapidly progressive, the amyloid isolated from rapidly progressive cases is progressing even in the transgenic system much faster. So where are the differences and similarities between prion diseases and Alzheimer's disease? So if we go down to the molecule level, I think that the induced misfolding, which means propagation from cell to cell, is known for both prion diseases as well as the Alzheimer's disease as a key mechanism. The conformation encoded strains, we know that they exist in both prion diseases as well as the Alzheimer's disease. The epigenetic phenotypic diversity, again, exists both ways in prion diseases as well as in the Alzheimer's disease. Where the difference starts is at the protein level. So if you look at the prion diseases, all the pathogenic protein is prion protein. SC stands for the scrapie form, which was the primordial first form studied. In Alzheimer's disease, it's amyloid beta and tau. Risk genes in the prion diseases is the prion protein gene itself. In Alzheimer's disease is the ApoE, apolipoprotein E cholesterol carrier protein. We know that in both diseases we have a sporadic forms, high frequency of sporadic forms than the genetic forms. We know that the mutant genes in prion diseases causing or linked to the prion disease or increasing the risk of the prion disease are all in the prion protein gene. In the Alzheimer's disease, it's the amyloid precursor protein, presenilin 1 and presenilin 2. This is the proteins responsible for the processing of that amyloid precursor protein. We know already that in the prion protein gene, as well as in the amyloid precursor protein, there are protective mutations. So which means that there are polymorphism in the gene which are protecting patients against the prion disease, as well as against the Alzheimer's disease. Familiar forms 10% for the prion diseases, around 10%, about 5% for the Alzheimer's disease. Now, obvious question is, if the prions are transmissible, and we know they are, is the Alzheimer's disease transmissible? And I think that until recently, the answer was no. This is a paper which was published by John Trojanovsky and Larry Schoenberger, CDC, using the iatrogenic cases of Creutzfeldt-Jakob disease. So we know that in all the iatrogenic cases of Creutzfeldt-Jakob disease, which were infected with prions through the growth hormone contaminated with CJD prion, that the pituitary glands contain also amyloid. Not only prions may contain prions, but also amyloid. So the obvious question at that first paper was, can we detect any higher risk of the Alzheimer's disease in the recipients of the growth hormone prepared from the cadaveric sources? The answer, looking at the autopsy records and clinical records, was no. So in effect, there was no higher frequency of Alzheimer's disease in recipients of the growth hormone in patients who effectively died from the Creutzfeldt-Jakob disease. So there was an infection by prions, but not apparently higher frequency of Alzheimer's disease. As it happened very often in science, when you look a little bit closer, and this is a paper which was published in Nature in 2015 by Brandner from Collinge's group, he found, when he actually looked at the brain tissue of those growth hormone recipients, that 4 of them had unexpectedly higher levels of the amyloid beta when you look at the age distribution. So when he compared those 4, and he had originally 8 cases, but if you look at the 4, they were accumulating much more amyloid than would you expect based on the controls at that age group. So as it happened again, in science we have 2 contradictory observations. So which one is right, which one is wrong? We initiated very quickly after discussing the issue with Larry Schoenberger from CDC, and we started to look at our US cases. And this is a work still in the progress. We can say that in a significant portion of those cases, we found that true we have higher accumulation of the amyloid beta in the plaques. We have also what is called vascular amyloid or amyloidopathy, and we have the also cases only with the mature plaques. So looking at the quantity of the amyloid in the iatrogenic CJD cases, patients who died because the CJD, because contaminated growth hormone, we have higher levels of both forms of the amyloid, 140 and 142. Those are two distinct forms. It's thought that the 142 is more toxic than the 140. Compared to the age-matched sporadic CJD controls, which were sporadic, they developed sporadic Creutzfeldt-Jakob disease. But you see there is a very significant increase in both forms of the amyloid. In some subset, in a small subset of the iatrogenic CJD cases, we also found elevated tau, specific form of the tau, pathological form of the tau in the hippocampus compared to the sporadic CJD age-matched controls in the hippocampus. The conclusion from this is that we see that the patients receiving contaminated growth hormone, contaminated with prions, also have higher frequency of amyloid disease pathology. Now, does it really mean that they have Alzheimer's disease induced by that growth hormone? I think that there are three questions we have to ask to investigate those possibilities and to understand what actually happened. I think that the higher frequency of the ID pathology, and this is, I think, one possibility, could be basically due to the fact that those patients received the growth hormone for reasons. I mean, they had some disease to start with. Many of them had surgeries of the pituitary glands. Many of them had trauma. They had radiation and other aspects. So we are looking with Larry Schoenberg and the NIH into those records of those patients and to see if maybe what happened before they actually received the growth hormone, maybe that's the reason for that higher amyloid frequency. The second possibility that we actually see the acceleration of Alzheimer's disease pathology by seeding effect of the amyloid beta in the contaminating cadaveric growth hormone preparations. We know that the amyloid is in the pituitary gland, so we know that it could be one possibility. So the way to look at it is we have now ultra-sensitive differentiation, which allows us to really trace back the amyloid beta and compare the cases and track back their origin. So the assumption is that all those iatrogenic sporadic CJD cases should receive the same amyloid preparation like they did in the CJD cases from the same source. The next possibility which we have to address is the cross-seeding of amyloid beta with pathogenic prion proteins. I mean, you know that in the test tube it may actually happen. So if you have an abnormally folded amyloid and you have the prion protein, they may change the conformation of each other. I mean, in some condition in the test tube it may happen. So in conclusion, I think that the story is still open and we have to keep working on it and to address all those three different possibilities. The fact is that the first paper I quoted, the Trojanovsky and Larry Schoenberger paper, clearly indicated that there was no Alzheimer's disease itself. So the symptomatology of Alzheimer's disease never really showed up. But maybe that is because the patient died through the Creutzfeldt-Jakob disease. If they would live long enough, maybe they would develop accelerated Alzheimer's disease. We don't have a really absolute answer to that. So this table just summarizes the other aspects, and I think that in general, some of the molecular aspects are shared by all those listed neurodegenerative diseases, specifically the induced misfolding of the proteins, deposit of different misfolded, misshaped proteins, high frequency of the, relatively high frequency of the amyloid, familiar forms, sporadic risk genes for polymorphisms related to the phenotypes, and epigenetic phenotypic diversity. So the conformational encoded strains, definitely in the Alzheimer's disease and also in the prion diseases. We don't know about Parkinsonism and frontotemporal dementia, but some of the in vitro studies indicate that it's definitely possible. So I think that in the future, the successful isolation and characterization of different conformers of A-beta and tau should help us to understand better the disease and also to develop ultrasensitive detection, which would be customizing the diagnostics and therapeutic approaches. To understand the trajectories of the disease we will hear about later in prion diseases as well as in the Alzheimer disease is absolutely essential for therapeutic intervention, because we saw yesterday in Richard Knight presentation how heterogeneous the group we are targeting is. So if we can identify cases and their trajectories early on, we can then differentiate therapeutic effect from no effect. And that's, I think, very important. These insights, I think, are very, very important for the therapeutic intervention. And I think all the therapeutic effective treatments, and we will again hear about it later in different scientific presentations, have effectively three different options. We can either increase the clearance or removal of the pathogenic conformers of the protein, or we can reduce the precursor protein, which is basically substrate for the conversion into the badly shaped toxic forms. Or we can inhibit the conversion of the precursor into the prion-like oligomeric states, and we will hear different approaches from next presenters. I would like to thank all the contributing co-authors, and I think there is a long list specifically from the Prion Center, as well as from the CDC, and I put the key players in all those projects I was listing today. The Center for Prions and Folding Diseases in Alberta with Jacques and Agnes and David Westerwey, Thomas Wisniewski and Eleanor Drummond at NYU. I think that all that research we are performing is possible only because support of the CJD Foundation and you and all families which are helping us with accumulating data and supporting us in all those investigations. And in the same time, I think that it's closing the circle in a way that when I see families and when I see patients, I think that it's very gratifying to know that what we do has real impact and has a real effect for real people. The work was supported by the grants from Center from CDC and NIH and Spitz Foundation. But again, I think that the most key is the CJD Foundation support. Thank you.